Hello, everybody. Um, can somebody just confirm it, please?
Yeah, you're audible. You're mute, Sridhar.
No, I know. Yeah, there you go. Hi, hello, everybody. It's my pleasure to introduce Miss Anjali Bajaj. Miss Anjali started her science career by joining the Bachelor of Science in Biochemistry at Daulatram College in the year 2011, which she successfully completed in 2014. And then she went on to do her master's in genetics at Delhi University South Campus in 2014 to 16, and then formally joined cleared CSR net examination and joined IGIB in August 2016 to pursue her PhD. This is her actual CV, the way chronologically it reads, but there are aspects to Anjali's character as well as her own personality, which many of you would have noticed and may not have noticed. But there's a small story to it. Why did I select her to the lab? Not many of you know. The reason was that she's also a very avid teacher. So now the questions that I had asked when she came around doing the rotations to select the supervisor was, tell me something that you did that you're proud about, or something that you think is very different from your colleagues. And the instant answer was, I have taught about a dozen plus kids. I used to take tuition for them in the science classes, and they all got very good marks in their 12th class and 10th class exam. And this is something that really stuck with me during the process of selection. If somebody has taught a 12th class kid, a dozen of them, and all of them getting good marks, really conveys the message that the fundamentals of the student was very clear. And that's something that I really appreciated. And that was something that I don't know if Anjali still knows about it. That is the reason why I selected her. But she was also the number one on the rank. So rather, she selected the lab and to work with me. So thank you, Anjali, for doing that. In terms of the time that she has spent here at IGIB, she has had 10 publications, and of that, 2 of them are really, I'm really proud that she could pull it through. And one, she will talk about it during her colloquium, which is part of her PhD work, which is her first author work. But what I'm going to talk about is the other one, which she rarely talks about. She managed to put together a publication which basically talks about how the Genomics for Understanding Rare Disease India Alliance Network really runs in terms of policy, education, research, the R&D, everything together. And it had 270 authors. So you can imagine working with that many authors, drafting a document, which actually is a white paper for how this 270 authors out of 70 or 80 clinical centers actually work together, was a huge amount of effort. And again, thank you, Anjali. She was the first author of that fantastic piece of work. And lastly, I think her core interests still remain, core interests beyond PhD still remain 2 things that she's very passionate about. And I know she will not want me to tell it, but I am going to tell that is still dance. So if you ever want to see a performance, tell Anjali, and she is always ready. And with that, I will let Anjali do her presentation. Good luck, Anjali. Go ahead, please.
Thank you so much, sir, for a very warm introduction. And I will now share my screen. I hope my screen is visible to everyone.
Yes, it is.
Okay, thank you. So, hello and welcome to my PhD colloquium today. The title of my talk is Genetics and Genomics of Cardiac Channelopathies in Indian Population. Now, before going into my work, I would like to bring your attention to these three prominent personalities that all of us would know. So there is a very misfortunate connect between all these three personalities. That is, that all of them passed away suddenly by a sudden cardiac arrest and without having any prior symptoms or diagnosis about it. And they passed away at a very younger age. Now, the stories of sudden cardiac death are actually very well recorded in even in cases of prominent athletes historically, because they are very common in athletes. And in fact, FIFA maintains a registry for sudden death that happens on the ground. And in that also we see that a trend of sudden unexpected death, that is SUD here mentioned here in the pie chart, increases more even when we talk about of younger people, that is age of less than 35 years. So here you can see SUD, which is very high whenever we talk about athletes, that is in the range of less than 35 years of age. So moving ahead, I'll just first start to talk about cellular basis of heart rhythm. So our heart is an electrical engine that keeps on beating throughout our lifetime, and there are many different kind of cells that are present in the heart, but the most prominent of them being cardiomyocyte, which actually give its rhythmic contraction and relaxation. Now on the cardiomyocyte membrane, if we see on the right hand side, this is the membrane, and outside is the extracellular space, and inside is the cytoplasm. So on the cardiomyocyte membrane, there are three different kind of ion channels that are present, that is potassium channels, sodium channels, and calcium channels. The orchestrated movement, opening and closing of these different ion channels actually leads to this characteristic membrane potential curve. And for each phase of this membrane potential curve, you can see different ions labeled on them. So basically, the opening and closing of different ion channels at each phase determines that at which stage the curve is it in. Now, this curve can be actually translated into the ECG pattern and can be read as ECG, that we know as P, QRS, and T waves. Now, P wave forms when atrial depolarization happens. QRS forms when ventricle depolarization happens, and T wave forms when ventricle repolarizes. So, going deeper into the context of ion channels, you can see here. So, this is the cardiomyocyte membrane that is depicted here, and there are certain ion channels that are present on the membrane of cells. So now, if you focus here closely on this ion channel complex, you can see that there are different kind of genes labeled on it. That is, for example, in this case, KCNQ1 and KCNH2. So each of these ion channels are actually macromolecular complex that is not only made up of a single protein, but are made up of different kind of proteins encoded by different genes. For example, here it is one of the gene, and then here it is a calcium channel that is marked in green, made up of CaCNA1C and CALM, and then there is a sodium channel here that is marked in purple, which is made up of SCN5A and SCNB genes. Now, understanding the genes and the proteins for ion channels, it is very important to actually know that how what happens if there are defects in these genes. So if you see, so minus and plus here means loss of function and gain of function. So loss and gain of function of different genes can actually result in the malfunctioning of the proteins that form the ion channels, and then that can be that can perturb the characteristic membrane potential curve that we saw previously. So red line here is the perturbed electrical membrane potential curve, and because of that, as a consequence, ECG also gets perturbed. So basically, if you see here in the red only, if you focus only on the red curve, you can see that the QT interval here is prolonged, and because of this prolongation, all the phenotypes arises that I'm going to talk about in the next slides. So now we understood that for maintaining the heart rhythmicity, cardiomyocytes and the membrane channels are important. Now, what would happen if the heart loses its rhythm? So then there is a problem because it will cause cardiac arrhythmia. So an arrhythmia is a problem with the rate or rhythm of the heartbeat. The heart can, during arrhythmia, heart can beat too fast, too slow, or with an irregular rhythm. So to understand the arrhythmias, the simplest classification is based on the heart rate. So if we see that the normal heart rate ranges between 60 to 100 beats per minute, and you can see the on the right hand side ECG pattern along with it. Now, if the heart if the heart beats too fast, that is, if the heart rate goes beyond 100 beats per minute, then we call it as tachyarrhythmias or tachycardias. And you can very well appreciate that now the ECG patterns changes drastically. Similarly, if the heart rate goes below 60 beats per minute, then it can lead to bradyarrhythmias or bradycardias. And again, you can see that there are change in ECG patterns. You will see complexes arising later in time. So this is the simplest classification of how arrhythmias happen and how do they look on ECG. But if we talk about causes of arrhythmias, especially in the young individuals like athletes we talked about earlier and other personalities, so genetics play a very important role in those kind of cases. And genetic causes of arrhythmias or the genetic types of arrhythmias are known as inherited primary arrhythmias or cardiac channelopathies. So cardiac channelopathies are of different types that are listed here, and each of these types have been associated with different number of genes in the literature. And the prevalence studies for these kind of cardiac channelopathies have been published for some of the disorders and in specific populations. So we can focus only on the top 4 kind of arrhythmias because I'll be talking more about them in my later slides. And also these 4 type of arrhythmias are mostly they are more reported, and genotype phenotype correlations are better documented for them. Now, talking about these 4 kind of arrhythmias, we see that there is a pattern that they follow, that there is a huge amount of genetic or genotypic heterogeneity between them. What does that mean? That means that for each kind of disorder, there are certain number of genes associated. It's not like simple one gene, one disease kind of scenario. For example, in long QT syndrome, we see there are 17 genes. And also you can appreciate that many genes are overlapping among different diseases. So to actually diagnose a particular type of syndrome, we really need to pinpoint the gene and the kind of variations they are causing. Because SCN5A, if it has gain of function variation, will fall into long QT syndrome kind of phenotype. And if it has gain of function kind of loss of function kind of variations, then it will fall into Brugada syndrome. So that's why the gene and the associated variation is very important to properly diagnose these different kind of syndromes. Now, if I only talk about long QT syndrome, then this is a kind of scenario it has. It has 17 different kind of genes, but not all of these genes are equally important in terms of their evidence and associations. There have been different classes that are reported in the literature that for example, 7 genes only are very strongly or definitive associated with the disorder. Out of these 7, KCNQ1, KCNH2, and SCN5A explains 75% of all LQTS cases. So now we can really focus on these 3 genes because we know that most of them they are explaining most of cases here. Next, talking about the phenotypic heterogeneity. Along with genotypic heterogeneity, it has phenotypic heterogeneity also. That means the symptoms of the arrhythmias range from asymptomatic to severe. So, for example, people can only suffer from chest palpitations, racing or lowered heart rate, shortness of breath, fatigue, dizziness, syncope, that is loss of consciousness, excessive sweating, or epileptic seizures. Or in severe cases, sudden cardiac death can be the first symptom. So this is very unfortunate. And these kind of sudden cardiac deaths can actually explain 10 to 30% of sudden unexpected deaths in young adults with no structural heart disease. So that's why this is very important for us, because these are actually treatable if they can be diagnosed in time. Also, under phenotypic heterogeneity, we see that because of different genes, there can be different phenotypes and different triggers. For example, exercise can be a trigger for arrhythmia, and that's why we see a lot of athletes are susceptible to them. So and for other disorder, it can be huge like loud noises, or for other or some other disorders, sleep can also be it can initiate during sleep. So that's why we need to actually dissect all of these genes based on on which the disorders are depending. So after knowing about genotypic and phenotypic heterogeneity, now we know that how important it is to document the genotypes and phenotypes all across different genes. So there have been few studies in the world that have documented really nicely the genotypes and phenotypes for the patients. For example, in this Italian study, there are 670 patients that are reported. In a USA study, there are like 541 patients that have been reported, and there are other studies also in which we see the representation of patients and dissection of genotypes and phenotypes. But if we talk about India, which we already know is so diverse in terms of its population architecture, only three different kind of studies have been reported, and that too with a very targeted approach. For example, in one study, only one gene was looked at. In other studies, only 2 or 3 genes were looked at. So, but we know that it is not only one gene or 3 genes that are important. There are number of genes and number of genetic heterogeneity that we need an account for. So that's why we identify this lacuna as scarcity of Indian data for these kind of disorders, and that's why it's important to study. And also, if we compare the Indian reported variations, whatever little variations have been reported as compared to the world variations. So basically, here these 3 kind of ion channels that you see are coded by the 3 major genes that I just showed, that explain 75% of the phenotype. If you see, all these small dots are the variations marked on these ion channel proteins, and if you have to really put an effort to actually see the red colored dots here, because only those represent Indian population. So this clearly indicates that there is a dearth of Indian data that still needs to come up to explain or to understand these kind of disorders in Indian population. So overall, summing up, the major challenges in the field are clinical diagnosis is difficult because we know that there is a heterogeneity. The clinician is also confused because it can range from asymptomatic to suddenly severe, such as sudden cardiac arrest. And many a times it can be misdiagnosed and confused with epilepsy, because epileptic seizures is also one of the symptoms. And because of this, genetic diagnosis is also difficult. As we have seen, there are many genes, and one need to really pinpoint and study all the genes to make a diagnosis. Also, there is incomplete penetrance and variable expressivity, again, in terms of the phenotypes. Only looking at the phenotypes, we cannot make out which syndrome is it. And the third major challenge is that there are very less population specific variations that are known about in Indian population, and because of which the knowledge about prevalent variations, we really don't know whether there are some variations that are prevalent in India, or if the disease at all is very prevalent in India. We didn't know about all of this information, which is very important to tackle the problem. And because of the above 3 challenges, the diagnosis driven treatment approaches are very limited, because we really don't know about the genotypes and phenotypes in Indian population. So based on these challenges, we have made the objectives as the first objective is the understanding the spectrum of genetic variants associated with cardiac channelopathies in Indian population. Second objective is developing a high throughput assay to screen prevalent channelopathy variations in Indian population. And third objective is the unraveling the genotype phenotype correlations of cardiac ion channelopathies in an Indian channelopathy cohort. That's a patient cohort. So I'll be talking about first 2 objectives first in a combined way. So for the first objective, we were really interested in looking at the genetic spectrum of cardiac ion channelopathy genes in Indian population. And for doing that, we didn't have any Indian specific baseline data that we could really use to look at those genes in which we are interested. So, but luckily, at the same time, our group did whole genome sequencing of 1,000 healthy Indians as the part of Indigen project. And this was a first of its kind study from India, which could actually actually document the baseline genetic data for Indian populations, and it has various aims, such as to enable clinicians and researchers in resolving allele frequencies of rare variations and genetic epidemiology of the diseases, and to improve diagnostic accuracy of genetic tests that are already running in India, and also to develop India-centric, cost-effective, affordable, and accessible genetic tests for public health applications. So the biggest surprise and biggest output of the study was that there were 55 plus million single nucleotide variations that were captured. Out of these 55 plus million variants, 18 plus million variants were actually novel or undiscovered till now, and that were there in Indian population. So we were really happy after this study, and we jumped into it, and we used this data to actually study the genetic spectrum for cardiac channelopathies. So these are the 4 disorders that we focused on. These are the majorly reported disorders, and there are separate genes that are associated with them. So basically, overall, for our study, we had to narrow down to a few set of genes. So we took all of these genes and narrowed down to 36 genes, primarily for 4 conditions. Now, taking these set of genes, we designed a pipeline. So we took 1029 indigenous data genomes for a VCF file, that is the variant calling file. We took all of these variants, and we annotated those variations using a tool that is ANNOVAR. And after annotations, we filtered the variations only for those genes which we are interested in. That was the list of 36 genes. And once we got those variations, we then filtered it for the rare variations, because we already know that the diseases that we are looking at are not very prevalent in the population, and we know that rare variations are have more chances to be pathogenic. So we only looked at rare variations first, and after that we looked at only the non-synonymous variations, because in many Mendelian cases, non-synonymous variations are could actually explain about 70 to 80% of the phenotypes. That's why we looked at non-synonymous variations. And after that, we used a computational prediction tool such as SIFT, Polyfer, and CAD to actually know about these non-synonymous variations, whether they are pathogenic or benign, so that we know what we are looking at, and we only took the variations that were predicted as pathogenic using these tools. Now, after looking at these after taking those variations, we used a database known as ClinVar. So ClinVar is a database that that documents all the clinically relevant variations. So we know that the variations we overlapped our variations with the ClinVar dataset, and we removed whatever was benign or likely benign variations in ClinVar, so that we are left with only the important ones which we are interested in. So after taking all these variations, we performed a series of classifications that is known as ACMG-MP classification. I'll be talking about this in the next slide. So after this, after doing ACMG-MP classification, whatever variations we got, only the high confidence PLP variations, that is pathogenic and likely pathogenic variations, we use for allele frequency comparisons. Now, in the second part of the pipeline, we only looked at the loss of function variations. So earlier I explained about the non-synonymous variations. Loss of function variations were separately looked at and annotated separately, because the tools that we were using were depending on the on VEP annotations. So again, we took only 36 genes, and we ran our tools to predict high confidence loss of function variations. So basically, loss of function variations includes frame shift variations, splicing variations, and nonsense variations that we were missing out in non-synonymous category. We covered here. And then we did ACMG-MP classification again, and then compared with different databases. So as the output of this pipeline, so basically we started with around 55 million variations, and out of after putting all of these filters, we ended up having 7 non-synonymous variations and 6 loss of function variations. So in total, we had 7 plus 6, 13 pathogenic and likely pathogenic variations. But the step before this is equally important, because we got 440 and 30 variations for which we did our ACMG ACMG AMP classification, that is 440 and 30 is 470. So this compendium of 470 ACMG AMP classified variations is a very important resource, because it can be used as a go-to resource for clinicians, because these are all Indian specific variations and have been classified with the updated guidelines. So talking about the ACMG AMP guidelines, so basically these are the guidelines that are based on different attributes such as population data sets, computational methods. So all of these different databases and literature screening it takes into account, and then on the basis of it we assign certain attributes to it. Based on the combination of the attributes that we assign, we classify the variation as pathogenic, likely pathogenic, variant of uncertain significance, likely benign, and benign. So basically we need to perform all the database studies and the literature studies separately for each variant, and based on the evidences we classify them. Now, talking about all the classified variations, we have classified all the 470 variations using ACMG AMP criteria. And on top, you see non-synonymous variations. So out of the total number of pathogenic and likely pathogenic non-synonymous variations were 7, and peel-off variations were 6. So that makes total up to 13 variations that we got. And the heat on the right side, you see a heat map in which on the x-axis you see all the genes labeled, and on the y-axis there are different classes: VUS, pathogenic, likely pathogenic, and likely benign. So basically, we concluded that we found high confidence pathogenic, likely pathogenic variations in definitive genes. That was expected, because these genes are really important. But also we found these pathogenic variations in limited or disputed genes. So basically, looking at a very targeted approach will not help. That means looking at only 1 or 2 genes will not help. Specifically in case of Indian scenario, we really need to look at all of the genes that are associated, so that we don't miss anything. After this, we compared the 13 pathogenic and likely pathogenic variations frequencies from indigenomes to other data sets here. So on the y-axis, all of the different population data sets are labeled, and on the x-axis, all of the variations, so 13 variations that we got, are labeled. And the size of the bubble you see in the bubble plot depends on how frequent is it is in particular population. And in few bubbles, you will notice outside circles. So these circles actually tells that that the variation is significantly different in that particular data set from indigenomes data set. So this is the bubble plot picture that is showing the significant differences of the variations that we found in indigenomes with the other population data sets. So we can notice that, for example, these 2 variations, these are only captured in indigenomes and are not represented in any other population data sets. So that's why indigenomes data set was really helpful for us, because it could uncover many other many pathogenic variations that were not there, not reported so far. And also we can see that NOMAD is said to be the biggest data set for comparison of population allele frequencies. You can see that there are many variations in NOMAD that had significantly different allele frequencies than indigen. So that's why this plot helped us to conclude that our variations are really important and really enriched in the indigenomes data. After this, we also looked at the unique variations across channelopathy genes. So basically, we had 470 ACMG AMP classified variations. We overlapped these variations with population data sets, and not only with population data sets, but other publicly available databases, and also the literature evidences. So after overlapping, what we found was that about 26.3% of variations, including 14 pathogenic and likely pathogenic variations, were unique only to the indigenomes dataset, and which suggested the importance of querying more and more Indian genomes, because still we think that we are missing a lot by only looking at the Western population data. So on the right hand side is the plot for the unique variations, and you can see that pathogenic and likely pathogenic variations are present in important genes also, such as KCNQ1, RYR2. And so this analysis was also important. After this, now we know that we had a total number of PLP variations were 13. So we actually went back to our Indigenomes data because remember, Indigenomes data is a data of 1029 individuals. So we mapped back these 13 variations, and we saw that there were 19 individuals or 19 alleles, or individuals is same in this case. 19 individuals were there having these 13 variations. And but if we only consider variations that are present in the definitive genes, having the definitive evidence for cause the disease, then we could narrow it down to 9 variations, and these 9 variations were present in 10 individuals in our data. So these two estimates actually led us to calculate the genotypic prevalence of cardiac and channelopathies in Indian population. So that came out to be around 1 in 103, or 1 in 1 in 54. So basically, on an average, it is 1%. So this high estimate, and keeping in mind that it is a healthy population that we are talking about, it's not the patient cohort, we could conclude that there could be two possibilities: either that the Indian population is more susceptible to channelopathy disorders. To support this data, we still need a larger data set. And second possibility could be that the variations that we have found, although they were pathogenic and likely pathogenic according to our classifications, maybe we don't know that they are common variations with small effects, and then there may be something else playing a role. So whatever the case may be, we right now we know that the disease prevalence or the genotypic prevalence of cardiac and channelopathies in Indian population is 1%. Now, taking this percentage to the translating this percentage at the country scale, we can estimate that taking account of Indian population currently consensus, we know that we are 140 crores strong, and if genotypic prevalence of these disorders is 1%, then we can see that there can be 14 crores people that can be at risk. If I put up the penetrance, incomplete penetrance also into account, then and calculate the numbers even very conservatively, it will come around as 5.6 crores. So basically, now we know after these estimates that we have a bigger problem in hand, that there are actually 5.6 crore people that can carry important variations or the pathogenic variations in these cardiac ion channelopathy genes that may predispose an individual to sudden cardiac death. So what can we do about it? So we first thing that came into our mind was to make an assay to screen these prevalent variations that we identified. So on the left hand side is the summary of the assay that we choose to design. So we already had a set of 13 variations, pathogenic and likely pathogenic variations from Indigen study. We designed primers for these 13 variations, and whatever amplicons we got, we first tested by Sanger sequencing that we could capture all of these variations. And then finally, after testing, we pooled our primer sets into two pools based on their compatibility. So we used different tools to do that work, and based on their compatibilities, we came up with two pools with 6 and 7 primer pairs each. And after using those pooled primers, we standardize a multiplex PCR assay, so that whenever we have to test these the presence of these 13 variations, we don't have to put separate reactions for each of the amplicon. So we designed a multiplex PCR assay approach, and after putting the PCR, we used we made the libraries using tagmentation approach, and then we sequenced using Illumina platform. Finally, we did the analysis. So basically, this is a sequencing based assay. In the end, it is not. It is unlike Sanger. It is very feasible and very time effective, because now you can screen multiple amplicons at once, and due to high coverage, because if we are sequencing it, the coverage is very high, and the confidence on the results is more, so there are less chances of a false negative kind of result. And next, it is very feasible in terms of cost and time to screen large number of samples together. And plus this assay carries all the advantages of a panel sequencing that we already know. Plus it can be used simultaneously with other disease panels. So in one particular flow cell, it is not a necessity that we put all cardiac channelopathy samples. We can actually pool these kind of samples with other disease panels also. So that actually increases its throughput even more. So when we tested using this approach, when we tested our primers with Sanger sequencing, so this is a Sanger validation that we got. So we already had indigene samples in hand in which we know the variations existed. So we just checked all of our primers that if they are working properly and if we can catch all of these variations. So we know once we know that our primers are working, we put up the sequencing run, and this is the result of that. So on the x-axis, you can see all the amplicons are labeled from 1 to 13 along with their respective genes, and on the y-axis you see that the mean coverage is labeled. So basically this figure has two parts to it. The top part is telling the mean coverage for each of the amplicon. So you can see that mean coverage is ranging from somewhere from 30x to 500 more than 500x. So the average on an average the coverage was 247x, and in the lower panel you can see a heat map figure. So in the heat map you can see that for each amplicon there were at least 94% of the bases that were covered at more than 10x coverage. So these two plots suggested that our panel is actually functional and it's working properly. So right now we are testing it on more samples, and again testing it on positive control like we did for Sanger to validate the panel so that we can start screening large number of individuals. So moving ahead for the conclusions and summary for this part. So our study serves as the starting point in the direction to understand genetic landscape of cardiac and channelopathy disorders in India, and to uncover unique as well as frequent variations in apparently healthy individuals. Second, we used ACMG-MP guidelines to classify variations, and we got in 13 pathogenic and likely pathogenic variations with the genotypic prevalence of around 1%. And important point is, as these disorders are treatable, our study is an important contribution towards public health in identifying high risk individuals with no apparent health problems as of now. And consequently, on top of it, we have developed a multiplex PCR assay, which will further aid in screening of prevalent variations. Now, after this, I am moving on to my next objective, that is objective 3, that is unraveling the genotype and phenotype correlations of cardiac and channelopathies in an Indian channelopathy cohort. So, moving on to my next objective. So basically, last objective was all about the healthy Indian population. This objective is more about the patient cohort. So as part of the Guardian Consortium, Guardian Initiative, we have built a channelopathy cohort in which we have included patients with a strong suspicion for inherited arrhythmias based on their clinical examinations, such as their ECG findings, symptoms of chest pain, dizziness, syncope, seizures, and a positive family history. So basically, this becomes a very important inclusion criteria. And when these criteria are met, only then a clinician sends or refers a channelopathy case for us to actually pinpoint the genetic diagnosis and to study the correlations and for novel discoveries. Next is a patient. If a patient is presented with sudden cardiac arrest, if it's a sudden cardiac arrest survivor, then also clinicians suspect it as a channelopathy disorder. So other inclusion criteria remain a consent form. A thoroughly filled consent form is required for actually start the study, and we also because this is a genetic study, so we cannot only discover a variation in the patient and leave it at that. We actually study the variation at least at the family level, and therefore we study at least trio samples for each variation or each finding that we get. And also strength of the genetics depends on the exhaustive clinical workup. So so we actually demand that the clinical workup should be very thorough, because only then we can make some sense of out of the genomic data also. So the workflow that we use is we first collect blood samples from the patients as well as the families, and then we isolate DNA, and then we use a whole exome sequencing approach using NGS platform to sequence patients' DNA only. And then we use bioinformatic analysis, and then we prepare the report based on then we report based on the findings that we get if it is positive. So moving ahead, so this is a review paper that we compiled, highlighting the Guardian initiatives and the status of genomics of rare genetic diseases in India, whatever has been reported. So if you are interested to know more about Guardian and for rare genetic diseases, you can go and have a look at it. Next is so we have the samples or the patient data that I'll be talking about in my next few slides is actually made the patient cohort is actually made in collaboration with a single tertiary center in India, that is AIIMS New Delhi, and the samples have been referred, the patients have been referred by a wonderful set of clinicians from AIIMS, and there were 53 probands which we have included in our study. So and which amounts to over total 200, more than 200 samples from patients as well as the family member samples. And the age of diagnosis actually varies from 7 months to 52 years. And provisional diagnosis for most of them was long QT syndrome, CPVT, Brugada syndrome, or other. So basically these three are the like main channelopathy diseases. And clinical symptoms at presentation varied from recurrent syncope, chest pain, episodes of shock, recurrent TDP, bradycardia, and others. So overall, if we see the number of probands, 30 were males and 23 were females, and the provisional diagnosis were mostly for most of the cases was LQTS and the related diseases. So on this, after getting the patient samples, we designed our approach as on the basis of differential clinical diagnosis. We included our patients and we did whole exome sequencing only for proband. After whole exome sequencing, we the reads that we get after sequencing, we mapped it back to the reference genome, and then we employed variation calling pipeline and prioritized variations. And after prioritizing variations, so basically variations have been prioritized based on their association with the channelopathy disorders, and sometimes you get only one variation in the end, or sometimes you get 4 to 5 variations in the end. So whatever variations you get, we have gotten, we did segregation analysis in the family samples by Sanger sequencing to actually establish that which of the variation is explaining the phenotype in the family. So the prioritization of variations is really interesting, because it is like finding a needle in the haystack. So for this, we employed another approach. So total variations, whatever we get from the whole exome sequencing, we again we employ a filtering criteria. We only look at deleterious exonic variations first and remove all the intronic and synonymous variations. And then we only looked at rare variations having minor allele frequency of less than 0.05, again because rare variations are more likely to be pathogenic. And then we looked at pathogenic variations predicted by the computational tools. And then from literature curation, we have compiled a bigger gene list for all the arrhythmia gene variations. And that bigger gene list is 240. So we looked at all of these genes to pinpoint to the genetic diagnosis. And next, if the clinician is very sure of the diagnosis, then we look at the specific channelopathy gene variations only. And then we use ACMG-MP criteria to classify these variations. We further look at the integrative genomics viewer analysis to see the coverage of our identified variation. Is it covered properly? And can we say can we pinpoint to the variation confidently? And finally, we look at these variations in a ethnically matched population. So earlier it was not available. Now we have indigenome data set. Whatever variations we get, we go back to indigenome data set and see if it has been covered or not. So using these pipelines, in a snapshot, I'll first would want to show the results. So initially we had 53 probands for which whole exome analysis and sequencing and analysis have been performed. And out of these out of these 53 probands, we could identify variations in 34 probands. And in 19 probands, we could not identify any variations so far. And in these 34 probands, the pathogenic variations were identified in 19 probands, and VUS were identified in 15 probands. So VUS are variations of uncertain significance. That means we still don't know whether they are very important, but because they lie in the associated genes, we had prioritized them and checked using Sanger. And out of these 15 VUS, 9 probands had novel VUS, which have been not reported so far, and 6 probands had reported VUS that have already been reported in literature. So this is the overall picture. And out of this, if we only talk about the variations, then the total number of variations are 39. In the brackets, you see unique variations. So basically, there were some variations that were present in more than one patient. So out of these 39 variations, pathogenic and likely pathogenic variations were 22 and VUS were 17. And as we already know that KCNQ1 and KCNH2 and SCN5A genes are very important genes carrying variations. They are definitive genes. We also see similar trend in our data that number of pathogenic variations were very high in KCNQ1 as compared to other genes. And 8 out of 17 VUS variations were present in limited or disputed genes. So this was the overall picture. And this is just a variation matrix plot. On the x-axis is the patient ID, and on the y-axis is the variations that are marked. So basically, this is just to give an overview that how many variations were novel and reported. And you see a lot of red dots and green dots. So all the red dots are pathogenic, likely pathogenic variations, and all the green dots have are variations of uncertain significance. So you can see, you can appreciate 50% of both the colors, and that's what we got. That out of the total number of cases, we could identify pathogenic and likely pathogenic variations in 36% of the cases, and VUS also in 36% cases. And the variations that we in the cases that we could not identify anything were 28%. And we plan to do whole genome sequencing for it, because we think that there may be some answers that are buried in there in the genome that we still could not figure out by doing exome sequencing. So out of this, so total genotype positivity was 36% for our patient cohort. And if we see only the definitive evidence genes such as KCNQ1, KCNH2, SCN5A, CASQ2, and RYR2, we see that these genes alone can explain 64% of all the identified variations. And while other limited or disputed evidence genes could account for 25% of the variations. So this was our overall snapshot. Now, because we studied 53 probands separately, and it's not possible to describe all of them. So I am taking one case study to show you that how do we go about doing the genetic study around a patient case. So on the left hand side, you can see a pedigree, and the arrow marked here refers to the proband, that is the patient because of which we started our genetic analysis. And you can see in the family that on the left hand side is a so in a pedigree, circle represents female and square represents male. So the proband had an elder sister who died suddenly with no unknown with an unknown cause, and the proband has a younger sister also and the parents. So we had samples for all of this family, and the proband was when proband was presented, he was 9 years old, and he had recurrent syncopal episodes. Syncopal meaning loss of consciousness episodes episodes since last 2 years, and the episodes usually occur after physical exertion such as playing, running, etc. And the proband was on beta blockers that are kind of medications given to the arrhythmic patients, and but still the symptoms persisted. So the clinician really wanted to do genetic diagnosis to understand why it is so severe. And the proband's mother was also on anti-epileptic drugs. Finally, the provisional diagnosis that we got was between LQTS and CPVT. That means the clinician was not very sure what is going on here, that why the severity is increasing as the proband is progressing with age. So we performed our pipeline for variation for whole exome sequencing of the proband and variation identification, and we could get a variation in a Cas-Q2 gene, which is a calcium regulatory gene. I'll be talking more about it in the next slide. So in this gene, and you can see here E239X is the variation, that is a nonsense variation, and so it is a homozygous variation that we found in the proband. And when we use our classification guidelines, it came out to be pathogenic. And on the right hand side, you can see the Sanger plots for all the family members. So on top, you can see there is a star mark peak, and only one peak is there. That means the proband was homozygous for this particular variation. That's why we see only one peak, whereas other family members were heterozygous for the variation. That's why you can appreciate two peaks at the same position. So we could provide a definitive genetic diagnosis for this family, that the homozygous variation was only present in the proband, and all the family members are heterozygous. And with that, we could also assure that the sister who was younger to the proband also carries the variation in the heterozygous form, and it really helped the family to screen the other members also. Now, talking about this condition, so basically Cas-Q2 gene and the condition. So CPVT is a condition that is caused due to Cas-Q2 gene, and it is very rare. The prevalence is estimated as 1 in 10,000, and mostly autosomal dominant forms are prevalent. That is more than 50% of the cases. We have an RYR2 gene that encodes for ryanodine receptor in the heart cells. It is responsible, but autosomal recessive is even more rarer, in less than so it happens in less than 5% of the cases. So the important point here is that it overlaps LQTS in terms of phenotypes, and it has a very high penetrance. It is mostly diagnosed at the young age following syncope or seizure episodes, but it can lead to life-threatening arrhythmias that occur especially after emotional or physical stress, like it happened in our proband also. And it is important to study because estimated mortality is 30 to 50% before 20 to 30 years of age if it left undiagnosed and untreated. So I looked at the literature, and we could find that there are already 104 patients reported with this condition, CPVT, in context of Cas-Q2 gene, and there were 65 unique variations that are reported. But important thing here was that there is no single Indian study with the genetic characterization of this disorder. So basically, CPVT is already rare, and Cas-Q2 gene is even rarer in this in the genetic picture of it that causes CPVT, and it is very fatal. So these all points prompted us to look at our data, and what we could find is very interesting. In our cohort only, we could find 3 cases of CPVT families, and all of them carried variations in Cas-Q2 gene. Now we already know from world data that RYR2 gene variations are more common, but we could get the rare variations, and that too in 3 cases. If you closely focus on the first and third row, you would find that in the first and third variations are same. So basically, we could identify 2 different families with the novel variations and the same variation. So basically, because of this, it prompted us to analyze if these 2 families are also related genetically. We did inheritance by descent analysis, but we could not get any findings related to that, which made us conclude that the 2 families were not related genetically. Hence, the two variations would have arisen independently. So basically, this was very important for us that we study more and more Indian patients also, because we really don't know which kind of genes and which kind of variations are more prevalent here as compared to the world data. So after this, so this slide is a very interesting closure for my last objective and this objective. That after studying all the Guardian cohort, what we did was we had 1,029 individuals from Indigen, and we had 13 PLP variations that we already found from Indigen. So these 13 PLP variations, after studying Guardian cohort, we overlapped with our patients' data, and we could get that there were three overlapping variations three overlapping variations between the Indigen and the Guardian cohort. So these three variations are listed here. So all of these variations were heterozygous in the Indigen data, but in the Guardian patient data, it was homozygous in one case and heterozygous in other two cases, explaining the phenotypes of the patients explaining the phenotypes of the patients respectively. So basically, these findings led us to conclude that both of our studies that were there, that they are very much related. In spite of doing first study in the healthy individuals, these findings underscore the clinical utility of our analysis of channelopathy variations in the healthy Indian population, and that prompts us to plan screening, large-scale screening programs and large-scale screening for all the at-risk people in the population. So after this, I would just conclude by giving the overall summary. So in the first objective, we used whole genome dataset, and we studied genetic spectrum of cardiac channelopathy disorders in self-declared healthy Indian individuals, which led us to estimate the genetic prevalence as 0.9 to 1.8% in Indian population. And as a part of second objective, we tried to translate that knowledge, whatever we got of prevalent cardiac channelopathy variations, into development of a high throughput screening assay to aid in reducing the public health burden. And in the third objective, we used whole exome sequencing data, and we studied genotype-phenotype correlations in cardiac channelopathy patient cohort, and we got a genotype positivity of 36%. And also, after the study, it prompted us to uncover many novel variations and uncover many different stories regarding Indian channelopathy patients, which we think we need to document more to get a clear picture on genotype-phenotype correlations in Indian population. So with this, I have made some future directions. So from Indigene study, we had all of the all individuals with we found all individuals who were genotype positive, but we are not sure that how many of them will become phenotype positive, because there is an issue of low penetrance in the disease, the probability of them being phenotype positive depends on many other factors other than the genotype also. There are many environmental and other factors. Similarly, in the other study, in the Guardian study, all of the variations that all of the patients that we got, we already know that they were phenotype positive, but we could not explain all of them using our genomic approaches or genetic approaches. And that's why this genotype positive circle is smaller here. And so that clearly indicates that there are still a lot of variations or genes that are still buried that we don't know about. So there is an issue of missing heritability. So taking these two studies into account, we really need to do a lot of work in this direction. So this finishes my future directions. Are there any questions?
Check the chat box. Read out the question.
Okay, so in the chat box, I could see only one question. It is from Sheetal ma'am. What is the genotypic prevalence of cardiac and channelopathies in other populations? So in other populations, also it is not very well documented. Like there are not many studies that have happened, like the study that we have done in healthy individuals. But there was one study in which the genotypic prevalence was reported to be 1 in 31. And it didn't use the rigorous ACMG AMP guideline approach like we did. It's a 2011 study. It reports 1 in 31. So only
Also,
that study.
I was keen to know the number of possible mutations that you have that may be pathogenic. How many of these are actually characterized functionally as detrimental?
So very few of them have been characterized functionally, and that's why we see in our data also that a lot of them fall under the category of variations of uncertain significance.
So that 1.9 to 1.8% estimate that you provide, that is for the ones that include the VUSs or without the?
Okay, I got your question. So the estimates that we had calculated, they are all pathogenic and likely pathogenic with functional studies reported in literature.
Okay, so that's
All
quite
of them
good.
have been, yeah.
Okay, all right.
Any other questions from the audience? This is the time to ask. Is there any questions from the audience? Please feel free to just unmute yourself and please ask the questions. Sheetal, if it's okay, let's go ahead with the ACSR presentation and we can take the questions at the end.
Sure, let's do that.
Yeah.
So moving on to my CSR 800 project. So the project that I did was to standardize medical genetic assay to enable better diagnosis. So disease that I worked on was mucopolysaccharidosis, which was caused MPS 3B. I'll talk more about it in the next slide. So mucopolysaccharidosis is a lysosomal storage disorder, which is characterized by accumulation of heparan sulfate. And it is MPS 3 is the most common form of mucopolysaccharidosis, with an estimated incidence is 1 in 70,000 newborns. And clinically, the patients have progressive neurodegeneration, behavior problems, mild skeletal changes. You can see coarse facial features and macrocephaly. And the clinical severity ranges from mild to severe. So the genetic basis of it is defined by the homozygous or compound heterozygous variations in the gene encoding N-alpha acetyl glucosaminidase, that is NAGLU gene. And the gene follows autosomal recessive pattern of inheritance. Now we selected this gene, and so this gene has 6 exons. The 6th exon is largest of all, and we made the primers for all these 6 exons, and there were 8 set of primers that we made, and we standardized all of these primers to make them into as an assay. So this is a gel image for all the standardized primers, and the specificity of the amplicons was confirmed by Sanger sequencing. And also we made a patient booklet for awareness and a protocol booklet for the assay, so that it can be run at a larger scale. So this was my CSIR 800, and I'll move on to my publications. This is the list of my list of publications. And if there are no questions, I can move to my acknowledgements.
Just a minute, Anjali. Let's just check. If anybody has a question, can you please unmute yourself and ask? I'm not hearing any questions, so go ahead, Anjali.
So I'll start my acknowledgement by thanking my supervisor, Dr. Sridhar Sivasubbu. Um, uh, so basically, from the very beginning, I really appreciate the freedom, the flexibility, and the support that he has provided to me. He has really allowed allowed me to work at my own pace and at my own time with my own interest, and that is very important. And he actually, when I joined, he actually given me many options in terms of projects. So there were many different projects that were running in lab, and he completely left that decision on me to take whichever project I liked in a broad landscape of the lab. And I really appreciate that I was given a lot of choices. And also so basically, I think I cannot thank him enough for how sensitively he has pushed me to, to take next steps in my career and to think about it very timely. And I really appreciate that support and guidance. Next, I want to thank my DAC members, Dr. Vinod Skaria, Dr. Chetna, and Dr. Shantanu Sengupta. So Vinod sir has also been really instrumental in all the work related discussions, and really helpful in all the work related discussions, and really supported always in his cool attitude, really supported to me for my work, saying that if you will not do, who will do? So that was a very important comment, that working in one particular area, it becomes our responsibility, that if the area is very important, we should do as much as we can do. We can contribute. And next, I'll move on to Dr. Chetna and Dr. Shantanu. So during my DAC meetings, they have been very curious and given very important comments for improving the work. And that was I really appreciate that. Next, I want to thank the directors that have been there. So in my span, there have been, in my tenure in IGIB, I've seen three different directors till now, and there have been many transitions. But the one thing that didn't change was the ecosystem. So I would really like to thank the directors for providing us a very good ecosystem to work here. Next, I would like to thank my collaborators at AIIMS. So all of the patient data that I have shown here would not be possible if we would not have such good collaborators in place already. So starting from Dr. Rajneesh Juneja, that who passed away 3 years ago, and his visionary leadership only made it possible to study cardiac channelopathy patients from a genetic perspective, especially in India. So that was very much appreciated. And other clinicians now include Dr. Nitish Nayak, Dr. Adipti Siddharthan, Dr. Gautam Sharma, Dr. Raghav Bansal, Dr. Rakesh Yadav, and Dr. Nitin Nayak and team. So basically, all of these clinicians have really, in spite of their very busy schedule, seeing patients at AIIMS, have really given important inputs and propelled the work forward. Next, I would like to thank the core NGS members, Dr. Shamsuddin, who actually led the foundation of the Guardian Consortium that we all work in today, and Dr. Ankit Verma, who initiated the cardiac channelopathy, who built the cardiac channelopathy cohort from the scratch. So he led the foundation for that. And now Dr. Rahul and Vigneshwar Senthiwal for helping with the indigenous work, and also all the Guardian related work that I have shown today. So their inputs were very valuable, and Vignesh, especially for the indigenous work, which we did together. I really appreciate it. Next is Dr. Mohammad Farooq and Dr. Uzma and Pooja Sharma. In their lab at Mall Road, it was really helpful for me to just go and do all the Sanger sequencing work. Without any hesitation. So it was very helpful for me. And next, I want to thank ACSR team, IGIB admin team, IGIB HR team, and IGIB IT team. So basically, these all teams remain in the background, but they really make possible all of that we do in their own ways. So they were very helpful and timely in conducting all of the important things that we need to do as part of our PhD. So huge thanks goes to them. And also, I would like to thank all the patients and their families for cooperating with us and trusting us with sending their samples and made the Guardian Consortium as it is today. So without their support, it would not have been possible. Next, I would like to thank IGIB for actually providing so much for the platform and for working for such working culture. I would be forever grateful to that. ACSIR for for the support for completion of our thesis, and CSIR for my fellowship, which I pursued for which I got for 5 years, and Guardian and Indigene as a whole, the teams and all the clinicians, all the scientists, all the collaborators that are associated with it with both the projects, because it was very helpful for that we could do all the work because of these two big projects that were there. And next, I will come to my lab. So basically, it is said that a person is an average of 10 persons of whom he is around. So basically, I was very lucky to have been around in the company of such amazing people at the VS and SSB at Sridhar Sir's labs and Vinod Sir's lab. So basically, there's a lot of interdependence that is there that we enjoy. No one works in isolation, and that part I really liked, because now we know that now I can actually tell about know about each of the person's strengths, and I can actually go up to any of them to talk. And I don't remember anyone with whom I have not worked in any capacity in the lab over the last few years. So I'm really grateful for that, and I've got to learn so many things from so many people here. And first of all, I would like to thank my seniors who were there when I joined the lab, and especially Dr. Ankit, who really helped me in the first 1.5 year of my PhD to strengthen my foothold in the lab, and to teach me how the lab works and how one should really move forward with the project. And next, Dr. Rahul, who is like an elder brother to me, and I just remember whenever I faint, he's there with a bottle of water. So that much good he is, and he's always open to discussions regarding anything. So that I really appreciate. And then Dr. Paras and Dr. Samata, who were really good in at one point of time, they were so active in conducting journal clubs and everything for labs, so that everyone improves and everyone learns together. So that I think was very impressive for me, that they are taking initiatives to actually do something for others, and to help the juniors as well. So I would really I'm really thankful for that. And then I would like to thank my wonderful set of juniors, Vishu, Srishti, Mohit, and Vignesh. So Vishu, I think I have known for the longest time, since my BSc, MSc, and now PhD. So the good thing about knowing someone for such a long time is that you know everything, all the strengths, all the weaknesses. So there are no clashes. You already know what to expect, what not to expect. And she and Srishti actually are two people whom I can go to at any time. If even if I whenever I have to discuss something very urgently, if I have a talk or something, even at night 10 p.m. 11 p.m. I can just ring them and tell them that come to Zoom, and they never say no. So that I really that much trust and that much flexibility I really enjoy with them, and especially for correcting all my slides, and every time I say that if so she is like my slide proofer, so she has to proof my all the slides before I present it to present them anywhere. So that was a lot of help for me, and I really thank her for that. Mohit also was with me during my MSc days. He was my batchmate, and now here also, and I really enjoy his company, and also his very critical comments for the work is what I really appreciate. Whenever so for my paper also, more than reviewer comments, he gave his comments before submitting. So that really helped me see all the loopholes earlier only. So that was very good. And I am really thankful to Vignesh. He actually speaks very less, but he mostly speaks on WhatsApp messages. So that also I appreciate. And I know that even if we don't talk for days or don't see each other every day, I know I have a there's a complete kind of trust that I can fall back to, and we know that we always want best out of each other. So that I very much appreciate. Other lab members, Mr. Gyan, Imran, and Hari, they are also have been very supportive and very they have been very good to be around in lab. And Dr. Anushri, Neha, Sandeep, and Deepak. So basically, they all are because of them, many things remain in a very organized fashion and closes very quickly. So for Dr. Anushri and Neha, especially for all the reimbursements and indents, we don't have to care about it anymore, because they are they are there to help always. And Sandeep and Deepak for all because we are working on so many patient samples and other indigenous samples, so all the management and everything that is there around that area, they manage it very nicely, and the work they are doing is very commendable. And in the VS lab, I would specifically like to thank Dr. Abhinav, Bani, Mercy, Arvind, Arushi, and Sahana. I have worked with all of them in several capacities, in doing file transfers for me, or helping with some plots, or working on I have also worked with GSA data with Arushi and Sahana that I have not included. It was a negative data, but I really enjoy working with all of them. And Mukta, Astha, Kavita, and Suruchi have been very nice friends also. So basically, overall, Vishu, Sashti, Bani, Arushi, Sahana, Mukta, Astha, Kavita, Mercy, everyone is they are like a happy pills of the lab. Even if you are feeling down or something, you can just go and talk to them. They will cheer you up. So that I really enjoy. And I have been lucky to work with trainees from whom I also got trained a lot. So Aviva, Venkat, and Biplop, they showed me that how we can what is the correct way of teaching certain things, and how we can make things how we should do things in batches so that nothing gets disturbed. So that was very important. And I also extend my gratitude towards Dr. Rachna and Dr. Ankita. They were in lab for very less time, but I think especially Dr. Rachna was so she used to speak very less, but whenever she speaks, she used to speak very solid, is what I felt. So there are few things that are still with me with whatever she said. So I'm very grateful for that. And all the Fish Lab members, so I'm very proud. I'm very proud to be a Fish Lab member, even if I've not included that part of my work here. It's a student-run facility, and the way the students run that facility is very amazing. And I think that is very commendable kind of collaboration that all of us show in Fish Lab. Next, I would like to thank my PhD batchmates for all of the fun activities that we have done during initial years of PhD, from organizing different events to choreographing dance sequences to everything. I really cherish that, especially with Dr. Samata here, who was my dancing partner also, and who actually used to push everyone to, so she would have a list of things that needed to be done before any event. So she usually used to push people to do everything. And Anurag, with the fantastic management capabilities, used to run all of the batch activities very nicely. And Khushbu, who was there, very supportive in the first few years of my PhD, and I really cherish that friendship. And other batchmates such as Salva, Madiha, Amit, Varun, Rohit, Shuvro, Surinder Singh Patel, and everyone. So all of them were very unique and very distinct, but it was good that we all could come together and do many great things together. So those times were very good. Next, I would like to thank my roommates, Ruchishri and Dr. Shipra. So IGIP hostel was for the first time I ever lived in the hostel, and these two have been amazing as roommates, and they actually taught me how to be a better roommate, and very warm and very welcoming. And I would really like to thank for all the nice time that we have spent together. And next, I would like to thank Anurag specifically. I think right now I have he's my best friend, and I don't know about 3 a.m. or 12 a.m. or any 24/7. He is there, and one thing, so everybody knows him that he's very good at managing stuff and very resourceful. But one thing that maybe few people will know is like is a good part of his personality is he's very calm, very composed all of the time. Like whether it is a big presentation or anything, whether it's my presentation, his presentation, whatever happens in the world, he's always very calm. So he has like Buddha sort of personality for me. I I still have to see the light in behind his head, but otherwise very positive, very calm all the time. I think his other name can be Hope. So if you have such kind of friend, I don't think you will feel low for a long period of time. So very helpful for last 4 years, especially for in my PhD. Whenever I had to discuss anything, he was already there with all his years and could guide me and could listen to me. First of all, that was very important. So I really cherish that friendship and that time we spent together. Last, I would like to acknowledge my family, my mother, father, and my brother. So I'm a very family oriented person, and I keep going back to them. And still, we are so basically, my family have shown commendable trust, commendable support, and commendable strength whenever I was away and I was here and I'm working. So they have always prioritized my career, work, or like everything related to me as their first preference. So that is very commendable and very supportive of them. Without them, I think I would not have lived away for such a long time in such ease and such pressure free. So that was very good for me. And this is a picture of me with my mother. So she has been very supportive, and from the initial age in itself, from a younger age itself, she has taught me that how self reliant and self independent is very important for women, and how I should not think otherwise. So I should so she has really helped me in my career also, in making my difficult decisions at times, in transitions, in everything. So she may not know all the scientific stuff, but she would try to listen to everything, understand it, and then explain it to all the relatives. So that is very good and very helpful. Lastly, I want to thank my sister that I never talk about. So basically, she left us 10 years ago, and we still, so she is still being missed. So becoming a doctor was a shared dream for her, for me, between her. So she had that dream that I should become a doctor and she should become a teacher. And now, if I look back for my last 6 years, I think I have done both. I have interacted with so many clinicians, and so I have taught to so many clinicians, and so it was all unplanned. But if I look back, I think it really completes a circle for me, and I think I had made. I have made her proud, a little at least. So she was my best well-wisher and my best critique also. So she is still being missed. Lastly, I would thank all of you for listening so patiently, and also I am a fan of quotes. So so I have only one thing at my desk, which is a quote book. And incidentally, for today, the quote was this: that you should laugh at your obstacles and cry at your success, and you should make challenge your friend and come out triumphant. So I really liked it. So chose to make as my last slide. So thank you, everyone.
Thank you, Anjali. Thank you very much for your nice presentation. And I hope you'll quickly write your thesis and complete the formalities from here onwards. Sheetal, any last words from your side before we conclude?
Congratulations. Good job.
Thank you, ma'am.
Okay. All right.
Yeah, congratulations, Anjali. Thank you, everybody.
Thank you.
Congratulations, Anjali.
Thank you.